Hi, it's Ms. Vitale. This podcast is on cells and corresponds to Chapter 7. Robert Hooke lived in the 1600s. He discovered and named the cell after the rooms where monks lived. When he looked at cork, which are dead plant cells, all he saw were the cell walls, and they reminded him of organized little rooms in a row. MATTHIUS SCHLEIDEN lived in the 1800s. He observed plant cells, and he came to the conclusion that all plants are made of plant cells. THEODORE SCHWANN in the 1800s also observed animal cells, and he came to the conclusion that all animals are made of animal cells. So Schleiden and Schwann are credited for developing the cell theory. The cell theory is a generalization about all living cells. It states that all living things are made of cells. Cells come from pre-existing cells, and cells are the basic unit of structure and function. There are a lot of different kinds of microscopes. The main type of microscope that you use in the class is a compound light microscope. Light microscopes let light pass through an object. It uses two lenses, the eyepiece and the objective lens. It can magnify up to 1,000 times. Electron microscopes use electrons to focus with a magnetic field. They can magnify up to 1,000,000 times. Scanning electron microscopes study the surface of the cells and give us a detailed 3D image of the surface. Transmission electron microscopes are used to study the inside parts of cells. We use these to study organelles and other internal cell structures. There are significant differences between plant and animal cells. If you look at the diagram of a plant cell versus an animal cell, do you notice any differences? Animal cells are irregular. Plant cells are square or rectangular and are arranged like bricks on a wall. The mitochondria is found in all animal and plant cells. Most plant cells have mitochondria, and only plant cells have chloroplasts. Animal cells do not. Animal cells also have small vacuoles throughout, and plant cells have a large center vacuole. Plant cells have a cell wall made of cellulose. Animal cells do not have a cell wall. We also classify cells as prokaryote or eukaryotes. A prokaryote cell tends to be much smaller than a eukaryote cell. Prokaryotes are usually unicellular. They only contain a floating loop of DNA in the cytoplasm. They do not have a membrane-bound nucleus. And the two main types of prokaryotes are bacteria and archaea. Eukaryotic cells are larger and more complex. Oftentimes, these make up multicellular organisms. The DNA, or the chromosomes, is contained in a membrane-bound nucleus. Plant, animal, fungi, and protists are the main types of eukaryotic cells. Organelle means little organs. These are small structures that organize the cell into different compartments. Each organelle has a specific structure and function. The structure and function relates to the organization theme. This is an example of many different types of organelles in a eukaryotic cell. We will talk about them individually. Cell parts are organized by what controls the cell, things that store, clean up, and support the cell, things that build proteins, organelles that capture and transform energy, cellular movement, and the different boundaries. A cell is like a candy factory. We're going to start with the nucleus, which is the control center of the cell. It contains the DNA. The nuclear envelope is the membrane surrounding the nucleus. It has pores in it which allow the movement of molecules through nuclear pores. The nucleolus is a dark spot on the nucleus, and it is where ribosomes are made. The cytoplasm is the entire region of the cell between the nucleus and the plasma membrane, or the cell membrane. It is where the organelles are suspended, and it basically has the consistency of a watery gel. There are lots of different organelles that store, clean up, and support the cell. The first are the vacuoles and vesicles. These are basically membrane-bound sacs that contain things like water or nutrients. In plant cells, there's a large central vacuole. In animal cells, there's many small vacuoles. Lysosomes are a type of vacuole that are for digestion. They contain digestive enzymes that break down materials. They can digest food or damaged organelles. They also can fight against bacteria and anything foreign that enters the cell. The cytoskeleton is a network that maintains the cell shape, helps to move the cell, or helps to move parts within the cell. Centrioles are areas that are only found in animal cells that organize cell division. However, their true function is questionable because if they are removed from an animal cell, the cell still divides, and plant cells never contain them, and they undergo the same type of cell division that animal cells undergo. Ribosomes are the organelle that builds proteins. They're one of the smallest organelles, and they are made of proteins and nucleic acids. They also are made of a specific type of RNA. They are built in the nucleolus of the nucleus, and then they are moved to either the rough endoplasmic reticulum or the cytoplasm. The main function of ribosomes is to build proteins. The endoplasmic reticulum is a network of folded membranes. If there's a lot of endoplasmic reticulum in a cell, the cell is very efficient. The rough endoplasmic reticulum is used to build proteins. Ribosomes are attached to it. The smooth endoplasmic reticulum is for processing carbohydrates and lipids. It does not have ribosomes attached to it. The Golgi apparatus works in conjunction with the ER. It is another network of membranes, and it's organized like a stack of sacks. Think of it as a stack of pita bread. It is found at the end of the ER. When products leave the ER, they move into the Golgi apparatus, which modifies, stores, packages, and transports the cell products. Chloroplasts and mitochondria are unique complex organisms. Both have folded membranes on the inside to increase efficiency. The chloroplast converts solar energy to the chemical energy stored in food, which is normally glucose, and it is the site of photosynthesis. The mitochondria converts chemical energy in food to a usable compound. It usually breaks down glucose and makes ATP. It is the site of cellular respiration, and oftentimes called the powerhouse of the cell. Cilia and Flagella are found on different types of cells, especially animal cells, and they are for movement. Flagella is usually a tail-like whip, and there may be one, two, or a few. Cilia are short and numerous and cover the cell and are for either moving the cell through water or moving food particles around the outside of the cell until they can enter the cell. Plant cells have a cell wall. The cell wall is made of cellulose, and it shapes, supports, and protects the cell. The cell membrane, also called the plasma membrane, is a lipid bilayer embedded with various different kinds of proteins, and it regulates and controls what goes in and out of the cell. The structure of the cell membrane, as I said, is conducive to its function. The phospholipid bilayer is two layers of phospholipids. A phospholipid contains a water-loving head and two water-repelling tails. The cell membrane or plasma membrane is selectively permeable, meaning that it allows some things in and out and not others. So how does material move through this bilayer? There are transport proteins that act as little conductors or conveyors for particles to move in and out of the cell. They can be passive where energy is not used, or they can be active where energy is used. Passive transport is a type of movement through the cell membrane that does not require energy. Passive transport is when particles move from a high concentration to a low concentration. Diffusion is the movement from an area where the concentration is a lot to where the concentration is low. Particles will move across a membrane. Facilitated diffusion is when particles use a protein to pass through, because in one way or another they cannot just simply pass through the cell membrane itself. Third type of passive transport is water. This also does not require energy. Osmosis is the diffusion of water through a selectively permeable membrane. Osmosis is when particles cannot diffuse through a membrane, so instead water moves from an area of low concentration to an area of high concentration, so it can dilute the higher concentration to equalize the lower concentration. When you have two different concentrations on either side of a membrane, the hypotonic solution is the lower concentration. The hypertonic solution is where the higher concentration is. When concentrations on either side of a membrane become equal, the solutions are considered isotonic, meaning that they both have the same concentration. Active transport requires energy. There are several different types of active transport. Usually things are moving against the concentration gradient. Protein pumps are small pumps made of proteins that transport things like sodium and potassium ions across the membrane of a cell. Endocytosis is when large particles, either liquid or solids, move into the cell. What happens is those particles force a pocket to form in the cell membrane. That pocket pinches off into a vesicle and carries the product into the cell wrapped in a membrane. Exocytosis is the opposite process. It's the same thing, but instead of a large particle moving into the cell, a large particle moves out of the cell. Both endocytosis and exocytosis require energy.